everybody, this is Mrs. Kraus, and in this video I'm going to go over the answer key for the mutations worksheet. So for question number 1, they ask you to name an amino acid that has more than one codon associated with it. So when I went to my codon chart here, I was able to see that the codons UCU, UCC, UCA, and UCG all code for the amino acid serine. So that's why I chose that one, although there are many other correct answers. Okay. Then they asked you to name an amino acid that has only one codon. So when I go back to my codon chart, actually my start codon, AUG, is the only one that codes for the amino acid methionine. And then the other one I identified was UGG, which codes for tryptophan. And again, there are no other codons that code for methionine or tryptophan. Okay. Then they ask you to look at the following sequence. The fat cat ate the rat. They ask you to take out the first H and then to group the letters again in groups of 3. But notice that when you do that, the sentence no longer makes sense. Okay, because we deleted a base, this is a frame shift mutation. It shifted how we read the sentence, and specifically a deletion mutation. Okay, so now let's take a look. This is our original DNA sequence, and I like to divide my paper up into sequences of 3 bases just because I know I'm going to use 3 RNA bases for my codon to determine my amino acid using the codon chart. So I went through and I wrote out the mRNA sequence that was going to be complementary to the DNA sequence, remembering that I need to use U's instead of T's in my mRNA. And then I used my codon chart and I wrote out the first 3 letters of each amino acid. Okay, and this is what I ended up with. My last one was a stop codon. Okay, then I have my first mutated DNA sequence. When I compare this DNA sequence to the original sequence, it looks like I had a substitution of a T for a C in the original sequence. This little symbol with the 2 arrows I'm using to represent a substitution. I know that this is a substitution and not an insertion or deletion because I have the same number of bases that I originally did, and the same groupings of 3. Okay, so for that reason, it's a point substitution, but we have to figure out which type of point substitution. My options are silent if it doesn't change the amino acid, missense if it does change the amino acid, and nonsense if it results in an early stop codon. And it looks like it changes the mRNA at this codon to UAG, which is a stop codon, so we would classify this as a nonsense mutation. Okay, with our second mutated DNA sequence, I see that I have added a G before my ACC here, and I can also tell that this is a frame shift because I have an extra base hanging off the end. Okay, and my groupings of 3 have changed. So when I go through and I transcribe it into mRNA and I translate it into amino acids, notice that all of the amino acids after that mutation have changed. Okay, so again, because I added a base, it's a frame shift insertion. Okay. Now I have mutated DNA sequence number 3. When I went through, I noticed that my mutation was a substitution of this adenine base, and in the original sequence, I believe it was a guanine base. And it changes my mRNA to AAU, but the amino acid doesn't change. It's still asparaginine. So I consider this a silent mutation because it doesn't affect the actual polypeptide sequence. Okay. For mutated DNA sequence number 4, I have another substitution. Notice that I end up with the same number of bases, the same groupings of 3. And in this case, a T was substituted where we had a G in the original sequence. And that causes us to have a slightly early stop codon. It's just missing one amino acid at the end. But because it does have that early stop codon, it's considered a nonsense mutation. And because it's a substitution, it's considered a point mutation. Okay, and then our last one, mutated DNA sequence number 5. When I looked at this compared to my original sequence, there was a deletion of a base. So I have GGA here. Golly, where is it? Okay, so it was, okay, second codon. Bear with me here. So it looks like, yeah, we took out the C here, so this becomes GGA. So there was a deletion of one base. I can also tell that because I end up with one fewer base than I had before. I only have 2 here instead of 3. And then when I write out my sequence of mRNA and amino acids, these 2 amino acids, proline and alanine, change after that mutation. Then I am asked to go through and translate all these mRNA sequences into amino acid sequences and identify which 2 are identical in terms of their amino acid sequences. And those would be mRNA-1 and mRNA-3. So this is just another example of that redundancy in the genetic code, where multiple codons can code for the same amino acid. All right, and that's it for this video.